All right, let's get right into it. For your CUET exam, some topics are just a total lock. They are guaranteed to show up. And believe me, this is one of them. We're about to go on a full-blown detective story to solve one of the biggest mysteries in biology. And you have to stick with me, because at the end, I'm giving you the exact blueprint to absolutely ace this section. So what if I told you that for the longest time, the smartest scientists on the planet were convinced that proteins, not DNA, carried our genetic code? Yeah, let's crack this case wide open.
So here they are, our two prime suspects. In one corner, you've got proteins. I mean, they're complex, they're versatile, they're made of 20 different amino acids. They seemed like the perfect candidate to carry a complicated code like life itself. And then the other corner, DNA. It was seen as, well, kind of boring. Just a simple, repetitive molecule with only 4 bases. Honestly, pretty much everyone was betting on protein. The big question was, who was the real culprit? Who was actually carrying the blueprint of life? This is a high-stakes topic for CUET, so stay sharp. You know, every good detective story kicks off with that first mysterious clue. And for us, that story begins way back in 1928 with a bacteriologist named Frederick Griffith. The funny thing is, he wasn't even looking for the genetic material. But his work with pneumonia-causing bacteria, well, he stumbled onto a piece of evidence that was about to change everything.
Okay, now what's so fascinating here is that final step. Let's just quickly walk through it. So Griffith had 2 strains of bacteria. A harmless one he called the R strain, and a deadly virulent one, the S strain. He injects the harmless R strain, mouse lives. Totally expected. He injects the deadly S strain, mouse dies. Simple enough, right? Then he heat kills the deadly S strain, injects it, and the mouse lives again. Okay, makes sense. But then, and this is the bombshell, he mixed the dead S strain with the harmless live R strain, and the mouse died. Whoa! Something from the dead bacteria had literally transformed the harmless one into a killer.
So here's the crucial takeaway from that. Griffith discovered what he called the transforming principle. He proved that some kind of chemical substance could actually change the characteristics of an organism and that this change was heritable. It was passed down. But he had no clue what that substance was. Now here's a massive CUET trap, so listen up. A common question will try to trick you into saying Griffith proved DNA was the genetic material. He did not. He just found the first major clue in our investigation.
So, for 16 long years, that clue just sat there, a total mystery. The case basically went cold. But then, in 1944, a new team of scientific detectives, Avery, MacLeod, and McCarty, they decided it was time to reopen the investigation and finally figure out what this mysterious transforming principle actually was.
And their method, which this table shows so well, was just brilliant. They used a process of elimination. They took that heat-killed S strain bacteria and used special enzymes to destroy one suspect at a time. First up, they used protease to destroy all the proteins. Did the transformation from R to S still happen? Yep. Okay, so protein wasn't the culprit. Then they used RNase to destroy RNA. Did transformation still happen? Yes, again. But then they used DNase, an enzyme that specifically chews up DNA. And look at that. Bam! The transformation stopped cold.
This was the moment the whole case just broke wide open. The investigation had finally zeroed in on a single culprit. By showing that transformation only stopped when DNA was destroyed, Avery, MacLeod, and McCarty could point their finger with confidence and say, DNA is the transforming principle. It's the genetic material. They had their suspect, but you know what? A lot of the scientific world still wasn't totally convinced. They needed one final, undeniable piece of proof.
So, for that final, irrefutable proof, we turn to the now legendary blender experiment. This was conducted in 1952 by Alfred Hershey and Martha Chase. And honestly, they designed such a beautifully simple and elegant experiment to provide that last piece of evidence that would close this case for good.
So check out their brilliant setup. They used a type of virus called a bacteriophage, which is basically this tiny little syringe that injects its genetic material into bacteria. In one batch of experiments, they tagged the virus's protein coat with radioactive sulfur-35. In the second batch, they tagged the virus's DNA with radioactive phosphorus-32. Now, why these specific elements? And that is an A+ question, because it's a huge point for CUET. Think about it. Proteins contain sulfur, but not phosphorus. And DNA contains phosphorus, but not sulfur. It was the perfect way to track each suspect separately.
And this right here, this is the smoking gun. After letting the viruses infect the bacteria, they literally used a kitchen blender to knock the virus coats off the outside of the cells. Then they checked to see what actually got inside the bacteria.
I mean, the result was just undeniable. Almost all of the radioactive phosphorus from the DNA was found inside the bacteria, and the radioactive sulfur from the protein, it was left outside.
And that was it. The verdict was in. This experiment proved beyond a shadow of a doubt that it is the DNA from the virus, not the protein, that enters the host cell to pass on the genetic instructions for making new viruses. DNA was officially, finally confirmed as the genetic material. Case closed. Okay, so now that we've convicted our culprit, let's take a second to understand why DNA is just so perfectly suited for this incredibly important job. What is it about this molecule that makes it the ideal blueprint for all of life? Well, it turns out any molecule hoping to be the genetic material has to meet these 4 really strict criteria. It has to be able to make copies of itself. It has to be stable enough not to just fall apart. It needs to be able to change, but slowly to allow for evolution. And finally, it has to be able to express itself, to direct the functions of the cell. And you know what? DNA ticks every single one of those boxes. RNA, by comparison, is less stable, which makes it a poor choice for most organisms, and proteins, well, they can't self-replicate. DNA is the clear, undisputed winner. All right, you followed the whole detective story? Amazing. Now, let's switch gears and lock this knowledge in for your exam. This next section is all about rapid-fire revision, and it's designed to make sure you can tackle any question CUE throws at you on this topic. This is where you can guarantee yourself full marks, so let's really focus in.
First thing, remember this timeline. It's the entire story in 3 simple steps. Griffith found the clue, Avery and his team identified the suspect, and Hershey and Chase provided the final proof. For your exam, just remember GAH. Griffith, Avery, Hershey. It's a super simple mnemonic that will keep the order perfectly straight in your head when you're under pressure.
Okay, listen up, because this is really important. Let's talk about the traps that CUT loves to set and how you're going to avoid them. Seriously, students lose marks on these exact points every single year. Trap 1, saying Griffith proved it was DNA. Wrong, he just found the principle. Trap 2, mixing up phosphorus 32 and sulfur 35. Just remember, P for phosphorus is in the phosphate backbone of DNA. Easy. Trap 3, claiming protein entered the cell. It didn't. And number 4, making a blanket statement that DNA is always the genetic material. Don't forget, some viruses, like HIV, actually use RNA. Knowing these traps can literally save you crucial marks. Don't fall for them.
So, let's end with this one powerful takeaway to lock it all in your brain for good. When you think of that final decisive experiment, just remember this. P is for phosphorus, and P is for proof. It was the phosphorus tag DNA that got inside the cell and provided the definitive proof that solved one of the greatest mysteries in the history of science. Now that you've followed all the clues and seen the final verdict, I'm actually curious, which of the experiments do you think was the most clever, and why? Let me know. Thinking it through will really solidify your understanding.